All right, good afternoon, CGIL. This week, we are very happy to have Dr. Ben Hayes, a professor at the University of Queensland, presenting for us. Professor Hayes has extensive research experience in genetic improvement of livestock, crop, pasture, and aquaculture species, with a focus on integration of genomic information into breeding programs, including leading many large-scale projects which have successfully implemented genomic technologies in livestock and cropping industries. An author of more than 300 journal papers, including in Nature Genetics, Nature Reviews Genetics, and Science, Professor Hayes also contributes to statistical methodology for genomic, microbiome, and metagenomic profile predictions, quantitative genetics, including knowledge of genetic mechanisms underlying complex traits, and development of bioinformatic pipelines for sequence analysis. Today, Professor Hayes will be presenting his research entitled Potential Approaches to Create Ultimate Genotypes in Livestock. So I'll turn it over to you now, Dr. Hayes, and you can get started.
Thank you very much. And what some of you may not know is I spent 6 months of my PhD at CGIL back in 1998, I think it was. And it was definitely the highlight of my PhD. So great to be talking to you today. So most of you, I assume, have heard about genomic selection. And today I'm going to discuss a different way of using all the data that we've accumulated with genomic selection. But just to touch on genomic selection, as many of you will know, the starting point for genomic selection are these reference populations where you have a large number of animals genotyped for genome-wide SNPs, 50,000, 700,000 SNPs, and they're also recorded for the trait that you want to improve. And I say large reference populations. Some of these reference populations are now extremely large. There was a paper just a few weeks ago in Genetic Selection Evolution, and that paper was using a reference population of more than 1 million animals. And in some of the genomic evaluations, using more than 1 million animals is now routine. And if you look around the world and count up all the animals that have been genotyped, you quickly come to numbers like 20 million. And genomic selection has had an impact on our livestock industries, particularly in dairy cattle, where it's been running the longest. And I could have chosen many examples, but here are just two. One from Australia, where we've been using genomic selection to improve fertility of the dairy cow herd. So in this graph, on the y-axis, you have year of birth, and on the x-axis, you have year of birth. On the y-axis, you have the fertility breeding value. And what you can see is after 2010, in the Holstein population in particular, the fertility breeding value has improved. And that was following a long decline in fertility, in fact, as a result of strong selection for milk production. Unfortunately, the genes that improve milk production have, on average, a detrimental effect on fertility. So fertility was going down, and we managed to turn that around by strong genomic selection on fertility. And dairy farmers have actually noticed that. They're reporting an increase in fertility in their herds. And just another example, in the US, genomic selection was implemented in 2008, and there was a very strong improvement in milk yield following the introduction of genomic selection. They also reported an improvement in fertility as well, as did many other countries. But the topic for today is really, is there an alternative way to use all this immense amount of genomic and, importantly, trait data that we've accumulated? And if you want to read about this approach that I'm going to discuss in more detail, it's just come out in Nature Genetics last week, I think it was. So the concept is, now that we have all this genomic data and phenotypic data, we can actually start to think about what's the ultimate genotype for a trait in our population. So with genomic selection, what we do is we calculate a genomic breeding value for each animal, say these 4 dairy sires, and for protein EBV, genomic breeding value, the first bull might have a breeding value of 8, the last one might have a breeding value of 3. So we would rank the bulls on their merit and select the first one for breeding. A different way of thinking about this problem of trying to maximize genetic gain, produce the ultimate genotype, is we can start to look into what chromosome segments each individual in the population carries and start to think about what would happen if we start to stack up or assemble the best chromosome segments at each position in the genome. So if you peer into the genomes of these animals, in this case, the blue are the desirable haplotypes, the best chunks of chromosome segment that increase protein at each position, and the red are the undesirable haplotypes. And what you notice in this example is even the bull that has the best genomic breeding value, he does carry a lot of good chromosome segments, but he also carries 1 or 2 bad chromosome segments. And the bulls that aren't so good on genomic breeding value, they might carry many poor chromosome segments, but just at a few locations, they may carry good chromosome segments. And what you really want to do is stack all those good chromosome segments together to assemble the ultimate genotype. So this is an alternative way of thinking of breeding, rather than just using truncation selection on genomic breeding value. We're deliberately stacking up these best or most desirable chromosome segments. And there's a bit of history to this, actually. If you look through the literature, there's a concept called gene pyramiding. And gene pyramiding was most often discussed in plants, but the idea was to stack the best alleles at multiple loci across the genome. And it actually goes right back to 1952. There were some Australian authors talking about rust resistance, but Frederic Hospital, who was a French geneticist, and also Jack Deckers talked about this quite extensively. The problem with gene pyramiding for complex traits is by focusing on just a few loci, you're only capturing a small proportion of the variance in the population. It works well when you've got alleles of very large effect, but that's typically not the case for the traits we're looking at. So this ultimate genotype concept is actually an extension of gene pyramiding, but taken for complex traits where you have mutations affecting those traits right across the genome. So we talk about quasi-infinitesimal traits, many mutations of small effect. This ultimate genotype concept extends gene pyramiding to this type of genetic architecture. And we've termed this optimal haplotype stacking. So to actually derive your ultimate genotype, to work out what your ultimate genotype is, the first thing you need to do is work out the effects of each of the chromosome segments in the population, those unique chromosome segments, and then stack them together. And we work out the effect of those chromosome segments through looking at marker haplotypes to track the chromosome segments, and then we work out the effect of those haplotypes in the population. So just peering into one chromosome segment here, the alleles, the marker alleles on that chromosome segment might be in the first case AT, CCCC, GTA, and we can estimate the effect of that haplotype by using the trait information, and we might come up with 0.2, whereas another haplotype has minus 0.1 and another haplotype still has 0.4. And then we can peer into our population and work out which animals actually carry the different haplotypes and start to think about assembling those chromosome segments. So again, in this case, Floss is a cow, dairy cow. She might have a stack of very good chromosome segments, very good haplotypes, so she has an excellent GBV. But again, just at a few points along her genome, she's carrying these undesirable segments, whereas Axel carries a lot of undesirable segments, so his GBV is poor, but he's got one segment that we want to stack into the ultimate genotype. So different authors have looked at the ultimate genotype in different species and how much extra performance you get over the current best individual. And when you do this, the gains that you can make are really quite astonishing. So the first people to do this, to my knowledge, were John Cole and Paul Van Reyden in the US. And when they stacked up the best chromosomes in the population in current dairy bulls, they saw that the dollar index net merit was 6 times greater than the best individual in the population. Catherine Kemper did this in the Australian population as well and got this 6-fold increase. In plants, it really depends on how diverse the population you're looking at, and the increases are anything from 1.2 up to 5-fold increase. So some quite dramatic increases are possible. The question is, can we get there? So how rapidly can we breed the ultimate genotype? If you think about the task of stacking all those haplotypes, all those chromosome segments, it's really going to depend on how much recombination you can get to stack up the desirable haplotypes into one individual. So it depends on the size of the chromosome blocks used to define your haplotypes, the number of haplotypes you've got within each block, which in turn depends on your effective population size, your genetic diversity, the number of crosses you can make per generation. The more crosses you can make, the more recombination, the more rapidly you can stack. The number of progeny per cross, which again drives your number of recombinations, and of course the recombination rate across the genome. So these are the parameters that really determine how rapidly you can stack and create your most desirable genotype, your ultimate genotype. Now, when Cole and Van Reyden published their paper back in 2011 and looked at the ultimate genotype, the way they defined chromosome blocks was whole chromosomes. So they literally worked out the chromosomes that every dairy bull in the population carried, every dairy bull that was genotyped, and stacked those best chromosomes together. And they came up with a net merit when they did that of $7,500, which is about 6 times the best bull in the population today. So quite a substantial increase. Pardon me. They could have actually gone even further than that by looking at just whole chromosomes. On a whole chromosome, there may be lots of desirable alleles, but there will also be some undesirable alleles. And to break out those desirable alleles and just stack those desirable alleles, you're going to have to look at smaller block sizes. So people do this in different ways, but a practical size to stack turns out to be about 1/4 of a chromosome. If you make the blocks too small, it just takes a very, very, very long time to stack up all the best blocks. Okay, we've also looked at the number of crosses per generation and the number of progeny per cross that you need or is useful to stack up these blocks. And I should point out that choosing the best parents to cross out of your whole population, the parents that contain most of the blocks that you want to stack, this is actually a highly combinatorial problem. So if you're going to choose 25 crosses out of 100 possible parents, it turns out that there are 8 by 10 to the power 51 possible crosses that you can make out of those 100 parents to choose those 25 crosses. That's just an enormous number. And you can't work through that number. You can't just calculate all those possible crosses. You have to have some optimization algorithm. So we use a genetic algorithm, which is a form of machine learning when we choose these crosses. Other people have chosen other types of artificial intelligence algorithm. But just to point out, this is a very large combinatorial problem. Nevertheless, you can do this. You can set up your genetic algorithm to choose these crosses. And I just want to show you now the effect of different numbers of crosses and different numbers, particularly different numbers of progeny. And this was, we did this in a simulated wheat breeding program, but really the answers are pretty general. They would apply across livestock as well. We used a wheat breeding program because we had founder wheat genotypes that we could feed into this. So Kira, our researcher, wrote a simulation program where she took 50 founder genotypes. She produced a large number of possible parents from those founder genotypes, and then she looked at how quickly could we get to the ultimate genotype when there were different numbers of progeny per cross. So this is what the results look like. And she compared the genetic gain to traditional truncation selection based on genomic breeding value. So what you have is different numbers of progeny per cross, 1, 10, or 100. And in this first graph, you have the number of breeding cycles, number of generations, and on the y-axis, you have the genomic breeding value. The yellow and orange colours are what you get with truncation selection, and the purple colours are what you get with this optimal haplotype selection, the stacking. What you can see is truncation selection actually does very well initially, but runs out of genetic diversity, so plateaus. Whereas the optimal haplotype selection, after a large number of generations, actually exceeds the genetic gain that you're making with truncation selection. But it does take a large number of generations to get there, sorry. And I'll come back to that. It turns out the best strategy that Kira found was to run a first generation of the stacking, so using the genetic algorithm to pick out the parents that had the best combination of haplotypes, and then to run truncation selection. And that's the blue line there. So that turned out to be the strategy that generated the most genetic gain most quickly. The interesting thing was, and I'll come back to this, was the optimal haplotype stacking, our stacking approach, maintained quite a bit more genetic diversity than the genomic selection, the traditional truncation selection on GBV, and I'll come back to that. This hybrid approach, where we first chose the parents that captured these optimal stack of haplotypes and then followed it with truncation selection to actually stack them up together, it turned out to look so attractive that we've implemented this in real breeding programs now. And this is in plant breeding programs. We haven't done this in livestock programs yet. But just to show you in the plant breeding programs, the way we did this was in a greenhouse where the light was manipulated to accelerate the breeding program. And by doing this accelerated breeding, we call it speed breeding, we could generate 7 generations of wheat in 1 year. And at the same time, we were running this stacking. And that turned out to be quite successful. When we took these stacked lines out to field trials, their yield was higher, substantially higher than check varieties. So just to show you that, we ran this program in 4 regions in Australia. And on the y-axis of these little graphs, you've got the yield of these wheat lines. On the x-axis is the different lines ranked. And the blue lines are our stack lines, and the red lines are the control varieties. So you can see we're getting quite a bit higher yield, which is pretty exciting. As I said, we haven't actually done this in livestock yet. It would be very interesting to do that. The problem is that you need large numbers of progeny per cross, per mating, to do this, and you need to turn over the generations pretty quick. Otherwise, it's not competitive with truncation selection. So in vitro fertilization is the way forward here. It is possible to have 100 offspring per mating if you do multiple pickups from a heifer, for example. So that is possible. And there are technologies coming through like in vitro meiosis. So still speculation, but looking more and more possible as these advanced reproductive technologies come on stream. Aquaculture is probably the place where you could really start to think about it, because most of our aquaculture species have so many progeny per cross, up to millions of eggs per mating. The problem then is you've got your cost of genotyping, but there are some nice ways of thinking about that through pooling and so on. Okay, at this point, I'm going to depart from reality a little bit and start really thinking about future technologies to build our ultimate genotype more quickly. The real constraint to doing this at the moment is we just don't get enough recombination quickly enough to stack these ultimate genotypes in any reasonable amount of time. So I'm going to consider 3 possible future pathways to getting to these ultimate genotypes more quickly. The first is accelerated and guided recombination, then direct editing of the ultimate sequence genotypes, and finally, just writing out the DNA of the ultimate genotype directly using synthetic biology. Just on accelerated and guided recombination, I said recombination's the main constraint here. Can we speed it up? It's worth remembering that recombination is actually heritable. You can select for increased recombination. This has been demonstrated in fruit flies, in worms as well, C. elegans, and multiple plant species. And I think it's been demonstrated in a fish species as well. So you could actually select to increase recombination. The other way of doing it these days is it's been demonstrated in a number of species that you can use gene editing to both speed up recombination, to drive recombination, and to target recombination exactly in the genome where you want it. The molecular technical complexity of this is still challenging, but the idea is there and has been demonstrated in tomato, for example. So what you could do is just speed up the rate of recombination by using gene editing. just to have more and more recombination. You have to be careful when you do this, however, that you don't break up your block of SNPs, your haplotypes, that you're actually using in your genomic predictions. Even in a method like GBLUP, we think about it using individual SNPs, but what it actually does is use combination of SNPs to track mutations, and it's that combination of SNPs that is used in the predictions. And if you break that up by recombination, you can actually reduce your genomic prediction accuracy. And there's a nice paper by Targan et al. Called "If It Ain't Broke, Don't Fix It," which clearly points this out. So what you want to do with your gene editing is target recombination exactly at the blocks you've used to define your haplotypes, so you don't break those haplotype combinations. And our researcher, Kira, again looked at this and asked, did it speed up the attainment of these ultimate genotypes? And in fact, it does. If you just compare the light blue line there to the dark blue line, the dark blue line is when you have this hybrid scheme of using choosing the parents that contain the best haplotypes and stacking them together using truncation selection. And the dark blue line is if you add in extra recombination. So I think 20 times extra recombination there. And you can see it does speed up your genetic gain, but it's really not fast enough. You're not getting you're not getting to that genetic gain many generations earlier. It's still too slow. So then we come to a different idea, which is just editing in the ultimate haplotype directly. And we have to say here that one of the main constraints with gene editing in livestock at the moment is it's been very difficult to actually identify causal mutations for complex traits. If you think about milk yield in dairy cattle, we actually have very few examples of actual causal mutations that we know affect milk yield. We've got QTL regions, GWAS hits, where we're very close, but we don't know the actual mutation. The other complication is for a trait like milk yield, there's many, many mutations affecting that trait. To use another complex trait as an example, the trait that's been most dissected, I guess, in most detail is human height. And there, the best evidence is there are about 7,000 mutations affecting that trait. So we'd have to do 7,000 edits across the genome. However, what we do know now, in some cases, is the sequence of the ultimate haplotype. So we know that there's a mutation somewhere in this region, this haplotype region. We don't know the actual mutation, but we can know the sequence, genome sequence of that haplotype through, for example, the 1,000 Bull Genomes Project, all the extensive sequencing people have done of our dairy bulls. So what we could do, rather than just focusing on one edit, we could just edit in that entire haplotype sequence. So we're doing a lot of editing of mutations that don't have an effect, but if we edit in the entire sequence haplotype, we will get that mutation. The question is, how many edits do we actually have to do? So another of our researchers, Christy, looked at this. She compared these best haplotypes to just an average haplotype in the population with the idea, if you had to edit in this haplotype into an animal, how many edits would you actually have to make? And it turns out to be a big number. Cattle are actually still quite diverse. So even in 100 kb haplotype, if you were editing into an Angus animal, the best haplotype in the Angus population, you'd be looking at 142 edits. Between our subspecies, So we work a lot in Brahman and Taurus cattle. If you had to edit in the best Angus haplotype into a Brahman animal, you're looking at something like 395 edits per haplotype, per 100 kb stretch. So if you multiply that up over the genome, it's a very large number of edits. You can save yourself a lot of editing by starting out with the best individual in the population and just editing out those few segments that it carries that aren't the most desirable, but it's still a lot of editing. There is another way of doing this, pardon me, and this is another approach which my colleague Tim Marnie came up with. You can actually clone out these chromosome segments that are desirable into what's called an artificial chromosome, and then just insert the entire artificial chromosome into your target individual. And by doing that, it turns out to be a lot more efficient than editing. So people have actually done this with things like the Bola region in cattle. So the the region that drives a lot of immune variation. They've taken that and, from humans, they've taken that region and put that into cattle. So there are actually examples of doing this. And if you think about just taking the best chromosome chunks, cloning them from each animal and putting them in using these artificial chromosomes, it's potentially feasible. Okay, the advantages of doing this, editing in these ultimate haplotypes, is that you've actually seen these sequences that you're editing in living individuals. You're not editing in something you've never seen in a living individual before. If you were worried about recessive lethals, for example, or deleterious effects, you could restrict your editing to only those haplotypes you've seen in a homozygous state. So you know they're not carrying lethal recessives. Still, I said at the start of this part of the talk, we're diverging from reality a bit here. I'd have to say this level of editing is not currently feasible. It's something for the future. The other approach that's starting to be interesting is synthetic biology. And what I mean by synthetic biology is just writing out these chromosomes, literally printing them out. And that sounds like science fiction, and to some extent it still is. But there was a paper last year which was pretty spectacular, where they printed out the genome of half of yeast, half of the chromosomes, inserted those into yeast cells, and demonstrated that they were functioning. So they literally printed out half the genome. There were issues with it. They had to do a lot of what's called debugging, which is fixing up parts of the genome, well, parts of the sequence that weren't quite correct, but they did manage to do it, and it was presented in this paper here. So this technology is coming along really quite quickly, something to keep an eye on. Again, not feasible in cattle right at the moment, but people certainly will be trying it, I think. Pardon me. Okay, so to conclude the talk, these ultimate genotypes look very attractive because they can have this performance up to 6-fold greater than the current best line or individual. I think the nice thing about this, this paper or this thought piece is that it marries up complex trait genetics and gene editing and synthetic biology. It gives those people working in gene editing and synthetic biology a bit of a target, something to actually target. A question you're probably all sitting there asking is, how realistic is this? How realistic is it to have individuals with 6-fold greater performance than the current best line or individual. And then you come to this discussion about selection limits. Do we see selection limits in our livestock and our crop species as well? And the answer is mixed, I guess. One of the answers is no, we don't really see selection limits. If you look at experiments like the Illinois corn experiment, where they've been selecting for percentage oil for more than 100 years, percentage oil has just increased linearly over that time. It's just gone up and up and up. And the fold increase is quite substantial, fourfold or so. In cattle, we've seen a 2 or 3-fold increase in milk production over the last 60 years. So we're not really seeing limits. What we do see limits to, though, in cattle particularly, and also we've seen this in chickens, is fitness. And if you think back to that first graph I showed, one of the first graphs on the decline in fertility that was related to the strong selection for milk production, that is a decline in fitness. So I think this is not just reinforcing that we definitely don't want to do single trait selection when we think about our ultimate genotypes. We really need to have fitness effects in there, fertility, survival, disease resistance, in an index that we're selecting on. The other point to make is that our ultimate genotype will change. I think it's a bit hubristic or egotistical to think we're going to know the ultimate genotype and it's never going to change. We're going to get better estimates of haplotypes over time, so that will change what we define as the ultimate genotype. There's new mutations coming through all the time. So part of this response that you see in this graph, on the slide, is due to new mutations. We're going to have more traits over time, like methane has become important in the past few years. And there's also changing environment, heat stress, for example, more disease challenges. And I've just talked about additive effects. There's also, of course, epistatic effects and dominance effects. We also, to meet this challenge that the ultimate genotype is going to change, you need to maintain diversity. And just to point out, and other people have pointed this out, and particularly the CGIL group has demonstrated that under strong genomic selection, we're seeing an increased rate of inbreeding. So this is under genomic selection, and this is Flavio Schenkel et al. Demonstrated this in the Canadian Holstein population, that the rate of inbreeding had increased. I guess one bit of good news is using this optimal haplotype stacking, at least on your way to the ultimate genotype, you do seem to preserve more genetic diversity. The reason for that is you're pulling in individuals that otherwise have a poor GBV but contain 1 or 2 good chromosome segments. And by doing that, you're pulling in a whole lot of extra chromosome segments into your breeding population. So if you look at the second graph there, the purple lines are from the stacking. It shows that you're preserving a bit more genetic diversity. And I think that's a very good thing. There's a paper we should all read that came out 7th of June, it looks like, in Animal Science by Ignacy and Daniela. I guess, highlighting or pointing out that we are starting to see some decline in fitness from potential decline in fitness from genomic selection. So this is something we should keep a very close eye on. So just to relate it back to the haplotype stacking, we should strongly consider fitness when we do this. Okay, I'll finish there, and just to say thank you to all my co-authors. There's a lot of people contributed to this effort, and thank you again for inviting me to give a seminar.
Thank you so much, Dr. Hayes, for your presentation and also for joining us so early in the morning over there in Australia. I'll open the floor now to any questions from people in the room or anyone online. We do have a question from Bruno in the room.
Hello, Dr. Reyes. First of all, congratulations for your talk. I'm very honored to listen a former CGILian delivering a so brilliant paper and presentation. Dr. Reyes, I would like to ask you about the inbreeding, because in traditional approach or truncated approach, we could have different individuals with different genotypes, but with the same GBV or equally high GBV. But in this approach of ultimate genotype, at least to my understanding, when you got the ultimate genotype, this genotype would be spread through the population. So how could this approach of ultimate genotype preserve more the diversity and be less sensitive to inbreeding than the traditional methods or the truncation selection? Thank you very much.
Yes, if you just take what I said very literally, you end up just having one genotype and everybody has the same genotype. Every cow would essentially be a clone. Obviously, that's a very bad idea, and we're not going to do that. The good thing about the stacking approach is on the way through to get to that genotype, you preserve more genetic diversity because you're pulling in these lines that you wouldn't use on GBV because their GBV is poor, but they contain a good chromosome segment. If you were actually going to do this, you'd have to think about it very carefully. You'd have to actually maintain a breeding population. It would be more like a plant breeding program where they maintain a breeding population and then the individuals that go out to the farmers' fields are genetically quite similar. But you would absolutely have to maintain that breeding population with the diversity. And you would, in reality, you wouldn't want to restrict the genetic diversity of your individuals going out to the farms too much. You would want to make use of some heterozygosity, for example, to improve their fitness. So they wouldn't be completely inbred. In the case of livestock, that would obviously be a bad idea.
Thank you very much.
Okay, and Flavio, you had your hand first.
Yes, I have a question. And in fact, two questions. But first of all, I'd like to say how good it is to see you, Ben. And thanks for accepting this and deliver this very, very interesting talk and food for thoughts, right?
Yeah, exactly. Yeah,
really nice. But I have a kind of a practical question for you. So you said that there was implemented in the breeding program of in plants. I don't remember the species that you mentioned, but it's implemented. So my question is related to the ultimate genotype for that breeding program was for what? Because you have to decide what to be the ultimate genotype. So if you can talk about that in practical use, if it was an index or few traits that were important for the crop. And the other one is that was not clear for me during the presentation is with respect to the variation of the selection response that you would expect with the stack approach versus the current approach of truncation selection. So have you compared the variation in selection response, right? Because that will be associated with the risk. Which approach is more risky in a breeding program? Can you?
I can actually answer that if you give me 2 seconds. I hadn't thought about that before, but we do actually have the answer. If I just Mm. Let me just share my screen again. Okay, so in the simulations at least, Kira did look at she's got the variation in these slides. So these are actually the 95% confidence intervals around the response. So I'm just looking at the variation compared to truncation selection. It's a good question, but I don't think it's that much greater than truncation selection.
Okay, Ben, we cannot see right now, but
oh, okay. I'll just tell you that it's about the same as truncation selection.
Okay, that's no different. That's
it's
important consideration. Was
yes, yes.
And how about this ultimate genotype for the crop? Was for what?
Yes,
was
so it was for yield of wheat. And we did that work with a breeding company. And it was strictly for yield. It's a bit interesting. And I see Mike Loheis is on the line, so he can comment too. But plant breeders typically don't use an index. They do a series of truncation selection on different traits. So the lines that went into the program, they would have been screened on disease resistance first, and only the surviving lines would go through to be evaluated for yield. So what we did was strictly on yield. We're now running a project to look at quality as well, so protein content and so on, and do a proper index on it.
Okay.
Yeah.
Thank you so much. I'll let other people to have a chance to ask you questions.
Yeah.
Thank you so much, Ben.
Thank you. Bayodi, do you want to ask your questions?
Yes, thank you. Thank you very much for the good presentation. I was thinking about playtropy. So let's assume for the ultimate genotypes, you have a very good animal. You have different haplotypes that have very fantastic. They actually influence a particular trait. What if they have a negative playtropic effect on other traits? How do we get rid of that?
Yep. So again, I think the best thing to do is to stack on an index where you take those negative effects into account as well. And what you're really looking for, ideally, is mutations that affect, just say, looking at two traits, both traits in a favourable direction. They would come up as very favourable haplotypes to stack. And in fact, in our wheat work, we've done that. We've looked at haplotypes that have effects on multiple disease traits, and some increase one disease, decrease another disease, but you can find haplotypes that improve resistance to both diseases, and they're really the ones you want to pull into your stack. Yep. So you can do that through an index. As long as you have full traits, of course, that's the absolute key, is to have a population where you've got those multiple traits recorded. That's really important.
Thank you.
Thank you. Okay, Mike, do you have a question?
Yes. Hey, Ben, nice to
Hi,
see
Mike.
you again.
Great to see you. Yep.
I do remember some of those fun times during your 6 months at CGIL. Yeah, I really intrigued by your talk, and one of the things maybe you could explain it again, why stacking isn't always better than truncation selection. I can understand why, the hybrid approach would be, have the highest EBVs in the shortest amount of time. But why would truncation selection outperform this? If you're able to identify the animals with the very best combinations of genes or haplotypes, why wouldn't that always be better?
I think ultimately it is always better, but it just takes so long to get there. So you've got so many generations where you're just trying to recombine chromosomes to get those best haplotypes or best chromosome segments stacked up. And particularly if the block sizes you consider are quite small, it just takes a very large number of recombinations to get there. And yeah, it just takes, if you don't have particularly large progeny sizes, family sizes, you're just looking at a very long number of generations.
Oh, I see. So that's why the fish example where you have so many eggs to select from, provided that you can genotype them all.
Yep.
The other question I had was, you mentioned fitness, and that's always a concern. How would you optimize both, your breeding goal that may not include all the fitness, things that you can't predict around fitness? Would you see like doing sort of an interleaved approach where you're doing the stacking one generation and then you're doing some combination whereby you're always checking fitness at some point in the intervals between? I'm just wondering if you had some thoughts about how do you preserve fitness. I really like the idea of what, preserving the best haplotypes, without truncation selection is going to get rid of the best haplotypes just by mistake. But you preserve the best ones in this approach. But how do you just preserve fitness? Have you thought about that?
Yeah, it's a great question. And since, Mehdi was here last year with Daniela, and Mehdi gave a presentation on inbreeding as a result of genomic selection and pointing out these declines in fitness that some people were reporting. So ever since then, I've been thinking about it. And I think, yeah, you have to have something like you're describing. So you run for a few generations, then generate progeny, check that they are fit, and then keep going as well. Otherwise, if you do it for too many generations, you're just going to accumulate deleterious mutations and you're going to have a problem, I think. But you could picture a breeding program where you're running your haplotype stacking, perhaps in vitro, but you're systematically spinning out animals that you actually take on farm and check that they are fit. And then if they're not fit, if you've got a major problem, then you go back to the stem cells that you've stored at that point in time and sort of start again. That's pretty radical, but something like that.
Yeah, cool.
I've been too influenced by plant breeding programs, Mike, where they do all kinds of things.
Yep, it kind of frees the mind a bit. Yep. Thanks, Ben.
Ricardo, do you have a question?
Yes. Hi. I have a question regarding the fitness as well. Some have the idea of having multiple breeding populations, one like totally weight on fitness, one totally weight on production, and then crossbreed, right? Would that be possible to have something in mind for the ultimate genotype there, that you have different ultimate genotypes and then have like a production crossbreed part of that?
Yeah, so one thing we're just starting to think about is actually this stacking with multiple traits. And the analogy of what you're saying is you could stack up all the good haplotypes for milk yield into one animal and all the good haplotypes for fitness really quickly, and then put them together in your commercial animal. And in the animal, it goes out to dairy farmers so that you have high yielding fit animals that you have on farm. You'd have to look at that, whether that's a better strategy than just selecting on the index. I'm interested in that idea of having the different populations. So do you know why people are proposing that instead of just an index?
Oh, it's just, I think it was in Denmark where they were like, oh, we could like between, but not between Holsteins and Holsteins, but Holstein and Jersey, right?
Oh, okay. Yep. And then make the cross.
But then it was just an idea of thinking, oh, could you look at it not as a breed or then also on weighting, but also on an ultimate genotype? But
no, it's an interesting idea. And I think one way you could tackle that is you define your ultimate genotype. These are the animals that you want to produce on farm, and you just let the computer decide. You let the machine learning tell you what the best breeding program to get there actually is. Interesting idea. Yep, that's good.
Right. Thank you. Are there any final questions in the room or online? No. Okay, I did have a question. I was just assuming we do see this implemented in livestock, we might see a very rapid response, like as you mentioned, but would it be possible to undo the selection of the ultimate genotype, or will mutation be enough to introduce more diversity to take advantage of?
Yeah, I think you want to always be taking advantage of new mutations. The challenge is, how do you actually do that? And people have written about this. With progeny testing, it was relatively easy to pick up those new mutations and pull them into your breeding program, because you're actually getting phenotypes on lots of those animals. With genomic selection, it's actually much harder. So you need to think about a strategy where you have some level of screening for those new mutations. And then when you find one, pull it into the ultimate genotype. But I must confess, I haven't thought about that in much detail, but it is something worth thinking about.
Okay, awesome. This was a very great discussion, I think. Thank you again for presenting for us today and for waking up so early or staying up so late, depending. But yeah, so I guess we'll wrap it up here. Thank you everybody for coming. The recording will be posted to YouTube and we'll give you more information on that later, Dr. Hayes. But yeah, thank you everyone for coming. We'll see you again next week. Thank you.
See you later.
Bye.